This is component 2 mock, Holy Cross mock, and it is mock number 4. And we're on to just question 1 to start with. Stage in cell division should instantly recognise this. Name the structures first. Well, start off with some easy ones. B and C, you should recognise those. C, centromere, B, chromatid. I'm going to put those in first. The whole thing is a chromosome with sister chromatids and a centromere. We've got this was the nucleus with a nuclear envelope around, so D is a nuclear envelope or nuclear membrane. So nuclear envelope or nuclear membrane. And A, well there's 2 of them and there's something beginning to form between them. So these are your centrioles. Name the phase shown. It can only be one, and what gives it away is the nuclear envelope is still there. It hasn't disappeared yet, so that's got to be prophase. So in all the other phases there's no nuclear envelope there, apart from the last phase, but then there'd be 2, so there's only 1. Type of division there. This is mitosis. Why? Well, we've got homologous chromosomes there, but they're not paired up. So these 2, possibly, but these 2 right far away from each other. So those are an homologous pair, but they're not paired up as bivalents, which they would be in meiosis. So no bivalence, or you could say no homologous pairing. State one biological function of this type of cell division. For example, repair of tissue, which will get you 1 mark, so you need to say 1, but I'll say a few. Growth, you're constantly growing. Replacing cells, asexual reproduction, lots of things you could say now. Let's put there, asexual reproduction. The diagram shows an early stage in this phase. The diagram drawn at the end of this phase would show several changes. Some easier than others to answer. What you need to do is go to your kind of knowledge about what happens in prophase, and you think, right, prophase, chromosomes become visible, the chromosomes shorten, condense, and this nuclear envelope disappears. You get the formation. You get the kind of centrioles migrating to the poles and formation of spindles. That's the sort of information that we want in there. Don't go on to the next phase and talk about chromosomes lining up at the equator yet, because it says later in this stage, so we're still in prophase. So, for example, the most obvious one would be the nuclear membrane breaks down. Centrioles moving. So moving to the poles. This spindle starting to form. And maybe further shortening and thickening, condensing these chromosomes, but they look pretty short and condensed as they are, but that would be an alternative answer as well. Is the cell shown an animal cell or a cell from a higher plant? And this just depends on whether you remember whether animals or plants have the centrioles or not. I can never remember, but it's an animal because the plants don't have centrioles. The higher plants do not have centrioles. So a fairly straightforward 13 marks, not much application, just AO1 remembering stuff really.